Good evening, everyone. Good afternoon, everyone. Good evening also for those who might not be joining from the time zone that I'm in, and good morning as well. Thank you for joining us today. My name is Dr. Lucy Machocho. I'm a technical programs assistant here at African Pharmaceutical Network, and I'll be your moderator for the day. I'm seeing we are currently at 44 numbers, and I'm kindly requesting that you give the rest of the participants a few more minutes, and then we can kick off at exactly 4 or 5 p.m. All right, so I see we're already 5 minutes into the top of the hour, and I'd like to welcome the new participants that have just joined. My name is Lucy Machocho. I'm a technical programs assistant here at African Pharmaceutical Network, and we are very happy to have you here with us today. I'll be supporting with the moderation, and I would like to encourage everyone that has just joined and also is here with us today. Could you please let us know in the chat who you are, where you're joining us from, what you do, and maybe share a link to your LinkedIn as well. We'd love to connect beyond this platform. And I'll walk you through the housekeeping rules for this session that will guide us throughout. So what to expect is we will start with an introduction to our work as African Pharmaceutical Network, and then go to the work that the International Society of Pharmacovigilance African Chapter does, before handing over the session to Dr. David, who will be taking us through the topic of genetic diversity in Africa and its potential impact on the future of pharmacovigilance and clinical trials. So with the grounding rules for the day, Please note that the session is recorded and this is for future purposes. The recording and the materials that will be used in the lecture will be shared in the session will be shared with you after the session. You will also note that your microphones and videos are turned off. That is to minimize any background noise and ensure that you have a smooth session. As mentioned, you can use the chat feature to let us know who you are, where you're joining us from, and a link to your LinkedIn where we can network and connect. And for asking questions, please use the Q&A section to ask any burning questions that you may have throughout the session, because the Q&A session is reserved till the tail end of the session. So in case any questions come up before the Q&A session, please feel free to post them in the Q&A section. And as we're engaging, please engage respectfully. In case you have any technical issues, my colleague is here and I'm also here to support. For feedback on this session or any other future sessions that you'd want us to have, please send us to the email secretariat@africanpharmanetwork.net. So I'll quickly walk you through what we do as an organization, that is African Pharmaceutical Network, and our reason for existing. So what we do basically is we focus on pharmaceutical system strengthening, and how we do this is through and bringing together all the stakeholders that are in the industry, that is from the professionals to the organizations that are working there, because we believe that these are our biggest resource in a continent that has limited resources. And for us, pharmaceutical system strengthening is not just ensuring that the African population has access to affordable health products, but also health products that are effectively address the unique diversity of the African population. And this is why we found this session to be very important to have. And one way in which we do this is, as mentioned, bringing together the stakeholders. And this is because these are the biggest resource in that they are the ones that are actually practicing on the ground. So they are the ones that best know what the continent needs, that is from what is working, what is not working, and why what is not working is not working. And they are the ones that can be able to effectively come up with solutions to understand why this is not working. And we ensure this is possible by having such sessions where we can share knowledge on such information. And this information is what will help us also shape policies that will ensure sustainability of the best practices that we'll have, best practices, and ensure that what is not working is actually working to cater to the needs of our patients. At the same time, it's through understanding what is happening on the ground that you can be able to nurture innovations and partnerships that will ensure that our patients are actually getting access to the quality, safe, effective, and health products and technologies. So the genesis of African Pharmaceutical Network began in 2020, and the reason why it came into existence was we noted some challenges in the pharmaceutical sector that we believed were worth addressing. That is one, the limited local manufacturing in Africa, which is still pertinent to date, with only very few countries being able to meet their pharmaceutical need. With most of us importing 70 to 90% of the health products that we use in our countries. Another issue that we noted is there was segregation across the sectors in the pharmaceutical industry, with people in the different sectors working in silos rather than collectively to understand how they can be able to effectively address the challenges that the sector was facing at the time, that is during the COVID-19 pandemic. And another reason was that there was no common platform to convene the stakeholders that were working in the industry and be able to talk about how we can make the future better. Our mission is to ensure reliable access to safe, effective, and quality health products and technologies in Africa. And we do this through several ways. This is pharmaceutical workforce development, business ecosystem support services, and policy shaping. The vision is also the same, that every African population, every African patient is able to get reliable access to quality, safe, and effective medicines and health products. These are some of our values. We have humanity, integrity, professionalism, client value creation, and teamwork. In our journey, as mentioned, we began in 2020, and during this time is when the foundational principles of the organizations were established. In 2021, we had the inaugural industry discussion forums, that is industry partners such as FKPM, PharmaConnect, and IFPME. In 2022, we signed partnerships for ensuring pharmaceutical workforce development with Empower School of Health, and also had the registration process officially initiated. And then in 2023, we established the Pharmacy Academy. We launched the first course, which is Regulatory Affairs, and also registered several members and had MOUs with IFPME, H3D, and Bloom Public Health. In 2024, we scaled up the courses in the Pharmacy Academy to incorporate pharmacovigilance and patient safety and pharmaceutical supply chain management. We also rolled out services such as advisory support and market research. In our services, we have 4 main buckets of services. We have the pharmaceutical workforce development. This is through the short courses that I've mentioned, and also the industry discussion forums such as this, where we share knowledge and ensure that professionals practicing in the field are equipped to practice the best practices that are currently in the market. Regulatory support services, that is ensuring regulatory support and pharmacovigilance oversight throughout the life cycle of the product. Business ecosystem support, we help unlock business value through market intelligence and also talent management through the pool of alumni that we have. Policy shaping and partnerships, that is to ensure that the policies that are actually there reflect the needs of the market, and how we do this is through sustainable partnerships and also advocacy. Who do we rely on to get to do our work? We have an advisory board of 3 members. That is John Paul Omolo, who serves as the board chair, Kirti Nasai, and McDonald Noswe. Our team is involved of the people that you see on the screen with you, in front of you. So we have Dr. David Odhiambo, who is our abled curator. We have Dr. Kelvin Odongo, who is our head of operations, Dr. Esther Anyango, our head of knowledge management, Dr. Maisiba Denish, head of business support services, and Dr. Anushri Patel, who is the Pharmacy Academy lead. And in the day-to-day operations, we have myself and Ms. Juliet Oma. We have a membership that is composed of individual and institutional, with the individuals having different rates for students and professionals, and the institutional having different rates for the African institutions that are domiciled in Africa and those that are founded out and domiciled in other countries but would want to have operations within Africa. Currently, we have enrollment for the pharmaceutical supply chain management course, and my colleague will be able to share the enrollment link for those who are interested. And one unique thing about our courses is that, like the industry discussions that we have, we usually ensure that the people that are talking, rather facilitating them, are practicing and are actually in tune with what is happening. So it's based on grounded experience, and also they are all for 3 months and very flexible to accommodate the busy schedule of professionals. Some of our partners, we have the ISoP Africa Chapter, who we are collaborating with today for this session, and they'll be making a presentation about their work in a few. And we also have IFPME, FIP, H3D, Bloom Public Health, the Pharmaceutical Society of Kenya, the Pharmaceutical Society of Zimbabwe, Empower Swiss, African Pharmaceutical Forum, Reculture, and the Spark. So how you can engage with us, you can either become a member, you can partner with us on projects, or you can learn with us. And with that, I'd like to hand over the session to George from the ISoP Africa Chapter to kindly take us through what they do in their work. George, over to you.
Yep. Thank you so much, Lucy, and good afternoon, good morning, colleagues. I'm trying to bring, can you see my screen, Lucy?
Yes, we can be able to see it.
Okay. I hope it works now.
Yes. Yes.
Beautiful. Today, no issues. So thank you so much for inviting ISoP Africa Chapter to make this presentation. I'm doing this on behalf of the Executive Committee. So I'll talk to you briefly I'll talk about the overview and the objectives of the chapter. I'll highlight on some key activities and initiatives of the chapter and upcoming events that the chapter will be organizing so that you'll participate. So to start with, the ISoP Africa Chapter is an independent nonprofit group of ISoP members. ISoP stands for International Society of Pharmacovigilance. Our members are within and outside Africa, though it's an African chapter. We do not limit the membership to only Africa. So we have a few members that are not residents on the continent. The chapter actually started in 2010 as ASoP, and ASoP means African Society of Pharmacovigilance in 2010. And a few years after operation, it became non-functional. So it was revamped in 2019 as the ISoP African Chapter. The current executive committee was inaugurated just about a year ago. The chapter is not registered in any country because the executive committee changes after every 3 or 4 years. Therefore, it's not a legal entity in any country. The objective of the chapter is to develop and foster pharmacovigilance in African countries. So we try to encourage and promote pharmacovigilance in African countries. As you may know, pharmacovigilance is a new science in Africa. The first 2 countries that joined the WHO program for international drug monitoring joined in 1992. So it's quite new on the continent. That's just about 25 years after the program became functional. So we support African countries to strengthen their pharmacovigilance system. We encourage and extend research in the field of pharmacovigilance and also to promote regular exchange of ideas in pharmacovigilance. Through meetings, symposia, workshop, we have a bulletin, and we have training programs specifically on Africa. And then also we organize our annual general meeting every year to promote pharmacovigilance and patient safety on the continent. So the map, you see the map of Africa or map of the whole world. As you can see, most of our members are from Africa. We have a little over 260 members as of this month today, from 32 countries in Africa, 29 countries on the continent, and 3 others are from Europe and the Americas. These are executive committee that was inaugurated in July 2024. We have Comfort, who is a Nigerian and is based in the USA. We have Helen, who is a Ugandan. Myself, I work in Ghana. I'm a Ghanaian. And Jayesh, who is a Kenyan, and he's our treasurer. Comfort is our president. The vice president is Helen. I am the secretary general, and treasurer is Jayesh. So what are the programs? What program do you organize? The chapter organized by, we actually meet every 2 weeks as a EC to discuss and to plan activities for the chapter. We also organize annual in-person meetings. The first one was held in July last year in Kampala. I'll tell you shortly about where the next one will be held. We also have a monthly newsletter that we use to engage members. We inform members about activities, what are the current issues in pharmacovigilance. We also published research work on a continent every month. So if you are involved in a research, kindly get in touch. We'll publish every research work that come out on the continent. That also promotes what you do out there. We have a directory of publication by members and other work that is done on the continent. Recently, we started an initiative we call the Spotlight, and this initiative is to highlight or elevate members' contribution to patient safety. We have profiled 3 members so far, and we hope to do more as the months goes by. So we'll do that. It's a monthly activity where we profile members and their contribution to patient safety on the continent. We have 3 active technical working groups. I'll talk to you briefly about the technical working groups and what they are doing. We have active social media platform. We have presence in LinkedIn, Google Meet, and then we also have some WhatsApp platforms, and also on X. We have collaboration with other chapters and institutions like the APN. So because of our collaboration with APN, that's why we are here today. So these are our TWG leads. We have 3 TWGs, as I mentioned. Professor Hanali Meyer and Dr. Mulugeta Rosum lead the regulatory and academia technical working group, and Kennedy and Christopher lead the public relation and communication technical working group, and Tuli and Adila, who also work with me at the FDA, lead the regulatory working group. So what are the activities that the TWGs has been engaged in over the past 1 year? The research and academia technical working group actually came out with a research agenda for the continent. They also have a mentorship program, and currently the proposal has been submitted for ethic approval, where they are doing a study on a signal management on Africa. They are looking at it's an exploratory study to find out to explore capacity barriers and facilitators for signal management in Africa. The practice and regulatory technical working group has been very active. It's actually the largest TWG. More than 50% of the members belong to this TWG. And the objective is to mobilize members. They've been involved in mobilizing members by having country focal persons. They have a few country focal persons. So if you are from a country that you are interested in joining the TWG, you could become a focal person if you are interested. They're also looking at ways of funding members to participate in our in-person meeting, particularly students. So we're looking for funding. If you are interested in supporting students in particular to take part in our meetings, you are welcome to do so. Because we believe the future belongs to the youth. So those who are in school, they will soon take over from us, and we need to groom them to do that. They are also looking for ways of interacting with the Vigilance Technical Committee of the AU3S program. The Public Relations and Communication Technical Working Group is the most active. They publish a monthly newsletter, and they also have a brochure that details our activities and what we do. Recently, we have 2 energetic ladies who are working to translate this in French. So our monthly newsletter and our brochures are in French now. We hope to translate them into other AU languages. They manage our social media platforms, and also they are involved in the spotlighting our members, as I mentioned earlier. So what are some of the activities that we have done so far? We have had 3 bi-monthly meetings so far. And these bi-monthly meetings, we invite experts who detail to us what they've been involved in, like we'll be hearing soon from David. We've had 3, one on MDA in Kenya, and I also presented on my PhD thesis that talks about the patient role in medication safety in Africa. And the last TWG we had was by Tariq, who made a presentation on navigating diverging perspectives, reasoning, evidence, and decision-making in drug safety. And our upcoming bi-monthly meeting is scheduled for the 12th of August, 2025. And we start our meetings at 9:30 and we close exactly at 10. So it's a short meeting. So I will invite you to take part in the next meeting. You can find a link on our social media platforms. The topic is nanomedicine and pharmacovigilance, transforming modern healthcare. And our speaker will be Teresa. So we have recently completed our strategy plan for 2024 to 2027. With input from members, we engage members at the Kampala meeting. We engage them through our online bi-monthly meetings and also a one-on-one. So it's a comprehensive strategy plan that details, that provide a clear roadmap which aligns with our goals and how we can achieve these goals up to 2027. So it's actually published on our website. So I will encourage you, if you're interested, just look at it and see what we want to do and where we are going in the next few years. So what are our future plans? We want to continue to enhance collaboration with other institutions. We want to actively participate at a global ISoP level. So I'll be talking to you about that soon. This year, we have suspended having our own, that's the Africa Chapter annual general meeting, because there is an ISoP meeting. The ISoP is a global meeting that's coming up in Cairo from August 24 to 27, 2025. So we feel it's important that we lend our maximum support to this meeting. So we are supporting ISoP Global to organize this meeting in Cairo in October. We will continue to elevate members' achievements and to actively participate in safety monitoring agenda setting by the African Medicine Agency. As you may know, AMA recently announced their CEO, which happened to be my boss. So we'll actively participate in the agenda setting by the AMA regarding pharmacovigilance. So at the ISoP meeting, the 24th ISoP annual general meeting in Cairo, we will have the ISoP Africa Chapter session. And we have a general meeting of the chapter. We also have a session that's about an hour session. So we intend to give regional updates from the African Medicine Agency, from our Pharmacovigilance framework of the AMA. I want to inform you that there are group discounts for people who want to participate in this meeting. And we've already sent an email to our members and the one they should contact if they are interested in the group discounts. If you have 5 participants, you have 5% discounts. If you 10 participants, you have 10% discount. So I will encourage those who are online and interested in taking part in the ISoP meeting in Cairo should take opportunity of this discount. 10% is a lot of discount that you can gain from this. So our 2026 ISoP Africa Chapter meeting will be in Namibia. Namibia is a beautiful country in Southern Africa, as you can see on the map. We had initial discussions with the Namibian team, and it's going to be May or June. The exact date will be announced soon, so please check our social media platforms. And maybe at the next APM meeting, we'll be here to tell you the exact date that the meeting will be organized. So we look forward to seeing all of you in Cairo and also in 2026 in Namibia. Just to mention that those who are members of ISoP, we have the advisory board elections coming up in August, September. It's going to be an online voting. All members will be sent an email. The ISoP advisory board provides strategic oversight and direction and governance for ISoP globally. We want the African voice to be represented. So the tenure of this new membership is from 2025 to 2028. So we are putting forward our president for election. So if you are a member, please look out and vote for Comfort Orga to be the next, to be an advisory board member of ISoP. So thank you so much for listening. And thank you, Lucy.
Thank you so much, George, for taking us through what ISoP Africa Chapter does. So quickly, in the interest of time, I want to introduce our speaker for the day. That is Dr. David Tusigomwe. And David is a postdoctoral fellow at the Sydney Brenner Institute for Molecular Bioscience at the University of Witwatersrand in Johannesburg, South Africa. Through various collaborations, David's research aims to address knowledge gaps regarding the pharmacogenomics landscape in Africa, and more broadly, the functional impact of novel and/or rare haplotypes across genes that encode proteins involved in drug metabolism, transport, and response. He is the developer of a diplotype calling algorithm, Stellar PGX, which facilitates calling pharmacogene star alleles from high throughput sequence datasets. David's research is supported by the Sidney Brenner Charitable Trust in partnership with the University of Edinburgh, and he is a current NHLBI Top Med Fellow under the mentorship of Professor Michelle Ramsey, Professor Scott Hazelhurst, and Professor Colin Masimirembwa. David, I'll hand over the session to you. Welcome.
Thank you so much, Lucy, and I would like to thank the APN and ISoP Africa for the invitation to present today. And my talk today will be about genetic diversity in Africa and its potential impact on the future of pharmacovigilance and clinical trials. So we know that in this audience, I think I don't have to talk much about this, but among patients taking the same dose of the same medication, we know that some will have side effects and others will have no response to the treatment. Of course, we expect most of the population to respond favorably, but this diversity that we see in response can be explained by a few factors, so including genetic variation, and that is the focus of my research. But we can see that you have other factors like the microbiome, which we are now beginning to also get an insight into how they impact drug response. So here on this slide at the top, I'm showing what we refer to as the one-size-fits-all model, which is currently in practice. Of course, we have some forms of precision medicine already taking place. So for example, if someone is a child, usually they get a lower dose of a medication. And if you're an adult, then you get a full dose. But we haven't yet, in many places, we haven't yet leveraged genetic diversity to optimize treatment response. But now in the era of sequencing and all this technology that we have, I think it is prime time now to be able to leverage this genetic variation and try to address some of the issues we see that are related to adverse drug reactions or treatment non-response. So you might find that in some individuals, you may need to reduce the dose of a medication, while in others, you need to increase the dose of the medication. And for others, you may need to choose a different medication altogether, depending on their genetic diversity. And this, of course, needs to be considered alongside the other factors that I showed on the slide before. So when we talk about pharmacogenomics, so this is understanding the relationship between genetic variation and drug response. But how do we do the testing, or what are some of the approaches to doing pharmacogenetic testing? So we see here that the best way is to do what we call preemptive testing. So this is where ideally for the patient, you have the genetic information or genetic profile already, and you apply some tools to predict how the patient might respond to the treatment. And then based on that, you can adjust the dose or choose another drug if the one you are going to give is predicted to lead to side effects based on the genetic variation. So that is the best approach, is to have the genetic information already. But we know that currently that is not practical in very many settings, especially in Africa. So there is what we now call the point of care tests. So this is where you test the you use technologies that you don't have to wait for a long time before you get the results. So it is still It still happens before you give the patient the medication usually, but then you have the technology in the hospital, in-house, and then you can make a decision based on the results. But majority of the times, what we know about genetic variation and drug response has actually resulted from what we call reactive testing. So in other words, you give the patient the medication, and then when you see a side effect, then you will ask the question, could this be due to genetics? And then you may send the DNA to a lab, and then they do the testing, and then you'll either find out that, okay, this may be the reason why the patient is not responding. So that is what has been happening for a lot of the discovery that we know about which variants lead to differences in drug response. So we can see that it's risky for patients if you want to do the reactive testing, but sometimes you can't help it because of the data sets. So in this slide, I just wanted to highlight some of the genes that I focus on a lot in my work, but it doesn't mean that these are the only genes that are involved in drug response. So we see, so since this is a pharmacy community, I don't have to talk much about drug disposition, but these are mainly the genes that encode proteins that are involved in the absorption, distribution, metabolism, and elimination of drugs from the body. So here we see genes like the cytochrome P450s, which are my favorites to analyze because they have very complex genetic architecture, and they are also involved in the metabolism of very many drugs, up to 75% of the drugs that are commonly prescribed. So the genes which are in the green here, these are the ones where there is a lot of evidence which shows that variation in those genes has been linked to drug response. And if you're designing a pharmacogenetic test, these are the genes that you would ideally want to have on your panel to begin with. Then we see other genes like the ones in the blue here. So these we refer to as extended ADME genes. And so these genes, they also have evidence of gene-drug interactions, but it's not as strong as the ones in the green. And then we have other genes that they may not necessarily be involved in absorption, distribution, metabolism, or elimination of drugs. But if you have variants in those genes, you may have some side effects because of having variation in those genes. So an example here is G6PD, whereby if you have G6PD deficiency and you are taking a drug like primaquine, you're at a risk of acute hemolytic anemia because then that primaquine and other quinolone medications, they will lead to an increase in the reactive oxygen species in the body. And if you're G6PD deficient, you won't be able to clear out those reactive oxygen species fast enough, and then it will lead to oxidative stress. So we see that there is value in understanding genetic variation in the context of drug response, and we need to leverage that so that we can improve patient responses to different medications. So when we talk about pharmacogenetic variation, what do we mean and how does this look like? So in the current pharmacogenomics community, we tend to use a shorthand where we refer to as these variations using a star allele system. This helps us to instantly understand what someone is talking about when they are referring to a variant or a group of variants that a patient may have. So for example, typically when we say star1 for a certain gene, we typically mean that the patient doesn't have any variant that may alter the response to a certain treatment, typically. It doesn't mean that they don't have variants in that gene, but it usually means that the variants they have are not going to change much in terms of the response. And actually, some people would refer to star1 as the wild type haplotype. And haplotype here means the group of variants that are inherited together from one parent. So we know that you inherit variants from your dad and from your mom. And then using CYP2D6 as an example here, so this star 17 is an allele that is very common in African populations, but as I'll show you later on in other slides, we don't see it in other global populations. So it is very interesting to study in the context of drug response. So we see that refers to this group of variants. And here in the functional impact, we see that having this group of variants leads you to have a decreased function cytochrome 2D6. And this is very relevant because cytochrome 2D6 breaks down about 20% of the medications that we take. So having star 17 is very relevant if you want to do precision dosing. Then you have other variants like this structure variant. So for example, you could have your CYP2D6 being deleted. But these are common in some genes and rare in other genes. In the cytochrome P450s, so these are very polymorphic, meaning that they have a lot of different variations. So we tend to see genes being deleted or even duplicated. So here, if you have a lot of copies of the gene and all the copies are functional, it means that you may have more of the enzyme compared to a person that has the normal gene arrangement. So this now brings me to talk about the high genetic diversity that we have in Africa. So this figure is from a paper that was published by colleagues from the H3 Africa Consortium. So H3 Africa stands for Human Heredity and Health in Africa. I'm sure many of you have heard of it and have seen some of the papers. So where they came together, and Lucy was talking about how for APN, you didn't want people working in silos, and you wanted everyone to come and start working together. So H3 Africa is really an exemplar of something like that, whereby all these researchers had data in their own labs, and then they came together, put that data together, and by analyzing it together, they were able to form a better picture of genetic diversity in Africa and what that meant for health and also migration. But today we won't talk about migration. So from the data sets that they analyzed, so these were whole genome sequence data sets at high coverage. So they were able to show on the right here, the population structure that we see across African populations. And what we see clearly is the diversity, as we can see different populations clustering separately. But we can also see a continuum. So this was also meant for us to really make out the continuum. So moving from Southern Africa, Central Africa, then West Africa and East Africa. So it really shows us that genetics in Africa also corresponds with geography. And that also means that when we are implementing this personalized medicine in Africa, we need to be careful not to take Africa as just one monolith, but we need to see what diversity is there in the different regions and then tailor our strategies based on that diversity. And then the question is, what does this mean for drug response? So in my work, I was able to take that same data that was analyzed by H3 Africa and also added other public data that we had available and looked at variation in cytochrome 2D6 mainly and also other pharmacogenes. And this work was done as part of a consortium that was funded by GSK and led by Professor Scott Hazelhurst, whom Lucy mentioned in my bio. And we did the variant calling and used tools to call those star alleles that I talked about. And for this, I developed a tool called Stellar PGX. So if you forget everything else from my talk, at least remember the name Stellar PGX, which is the tool that we developed for calling star alleles from genetic data sets and looking at variation in those genes that are involved in drug response. The other thing we did was validating some of the novel star alleles. So we know that when you're working with African data, because a lot of African populations have been underrepresented in research to date, you're more likely to find novel alleles that may or may not be relevant for drug response or disease, depending on what you're looking at. So this is just a snapshot of the findings from my own work and from the work of our institute in general regarding variation that may impact drug response. So the first figure here shows us the discovery curves of variation in these genes. So what is seen at the bottom here in the pink, this is for European populations. And we can see that when you could compare with the variation in Africa, which is in the blue and the green, we see that variation in Africa is far greater than variation in Europe. And the data for the European populations here is slightly low coverage in terms of the genetics because of the technology that was used. And then the data that is in green for Africa is also low coverage. So we can see that even when you look at low coverage data, Africa is still more diverse than Europe. And then the data in the blue is the higher coverage. What we mean by higher coverage is that we sequenced deeper in those genomes, and we can see that you pick up way more variation in Africa. And this just shows us why we need to include African genomes in research. And then on the right, here, I just wanted to highlight what we found as high impact variants. So in other words, after we did our variant calling, we wanted to know which of these variants in these ADME genes are more likely to be functionally relevant. So we used a set of bioinformatics tools to figure out those high impact variants. And what we are seeing here is when you look at Southern Africa, South Central Africa, Far West Africa, and Western Africa, we see that they shared about 104 high impact variants. But what was striking to us was that each of these different regions had quite a considerable number of variants that were not observed in the other regions. So for example, here we see 133 variants in Southern Africa that are not observed in the other regions in Africa. So that also shows that you can't just treat Africa as a monolith when you're considering these precision medicine strategies. Then this brings me to some examples of, why genetic variation in Africa matters in the context of drug response. Just examples that we can all relate to. So the first example here is with cytochrome 2b6 and efavirenz. So many of you know efavirenz as a drug that has been part of antiretroviral treatment regimens for years, but many African patients were experiencing neuropsychiatric side effects to efavirenz. And the question was why? So it turned out that a lot of African patients were poor metabolizers of efavirenz, and that's shown here at the top. We see in the yellow here that that there's a disproportionate distribution of poor metabolizers when you look at Africans versus European participants. So, and because of that, they were not clearing efavirenz fast enough, and that that meant that they had the high exposure to efavirenz for a prolonged period of time, and that was now leading to those neuropsychiatric side effects. We see also that we have a high proportion of intermediate metabolizers in Africans. That is this IM. So both PM and IM would be at a risk of those side effects for efavirenz. Then the question was, why are we seeing this disparity in the distributions of phenotypes for response to efavirenz? So it turned out that in Africa, we have a high frequency of alleles that are either not observed in other populations or they are more enriched in Africa compared to the other populations. So we see here in the red, this allele called star 18. As I showed earlier, whenever I show a star allele like star 18, it means it is a shorthand to refer to a group of variants that are inherited together from one parent or the other. So here, this allele is a non-functional allele, and we see it is common in Africa and very, very rare or not even present in the other global populations. Then also there is this allele, star 6, which many of you probably know. So when you combine the high frequency of this star 6, which is also a decreased function allele, with the 18, we see that it will lead to a high proportion of Africans that are not responding well to efavirenz, well, that are not metabolizing efavirenz fast enough. So, of course, now we know that there are newer drugs for HIV treatment like dolutegravir and cabotegravir, but we are also starting to see some side effects to those medications. And genetic variation definitely may be playing a role as well. But this shows us the importance of studying genetics in Africa and in the context of drug response. So, yeah. So then we know that when people were switching from efavirenz to dolutegravir, there was this observation of weight gain. And the question was, why is there weight gain? Is it because the new drug dolutegravir is the one causing the weight gain? So some groups have shown that actually those alleles that were toxic, that were leading to increased toxicity from efavirenz, when you now switch to another drug, you reduce that toxicity, and then that might then contribute to that weight gain. But it's still a very interesting discussion about why we are seeing things like hyperglycemia with dolutegravir and dyslipidemia as well. Yeah. So the second example that I would like to talk about is cytochrome 3F5 and tacrolimus. So tacrolimus is a very interesting medication. It's a medication that they give to prevent organ rejection. So it is beneficial for a patient to have tacrolimus for as long as possible so that they don't reject the new organ or the new tissue. So what we see in Europe is that for most of the participants, they don't have an active form of this gene 3F5 because they have this a high frequency of this allele called star 3, which has no function. So this is actually beneficial because it means that many European patients, when you give them tacrolimus, which is already in the active form, they are not going to break it down. So they are going to have a high exposure to tacrolimus for a long time, which in this case is actually beneficial rather than the context of efavirenz. So when you now come to African patients, we see that a lot of Africans don't actually have this same allele, the star 3. So the star 3 is relatively less frequent in Africa, and actually a lot of Africans have a very active 3F5. What this means is that a lot of Africans are expressors of 3F5, and they will metabolize the drug quickly. And this is not good if you want to prevent organ rejection. So this would mean that you would have to increase the dose of tacrolimus that you give to majority of African patients. Whereas for efavirenz, you would have had to actually reduce the dose. Here, you would have to increase the dose because of the drug-gene interaction. And then I also wanted to highlight work on 2D6 just because it metabolizes so many medications. So I just wanted to show you the data that I promised of the STAR17 being very common in Africans, but very rare in other populations. And we have STAR29 as well, that is common in Africans. And these two alleles decrease the function of CYP2D6. The relevance for this has been shown for the drug tamoxifen, which is used to treat breast cancer. So a lot of African patients carrying those two alleles would require higher doses of tamoxifen for it to be effective. So we see different scenarios here where you either have to reduce the dose to prevent adverse drug reactions or increase the dose for a drug to be effective based on genetic variation. And so this is really what intrigues me about this work, and I think why we should all be interested in leveraging genomic diversity to enhance drug safety and efficacy in African settings. So the other thing I wanted to talk about is that, yes, there is diversity between Africa and Europe. And the reason why I compare a lot to Europe is because a lot of clinical trials involve mostly European populations. Most of the clinical trials, I mean, very few clinical trials are being done in Africa because of various reasons. And that, of course, is one of the reasons why we are seeing that because of not accounting for this genetic diversity that we see in Africa, that is resulting in a lot of patients not benefiting from the current, the standard regimen, or needing dosage adjustments. So this also shows that when we are, for the new drugs, we need to take that into account. But in this slide, I would also want to highlight that there is also diversity within Africa. So like here, I'm showing the distribution of alleles in cytochrome 2D6 across different populations. And we see that they don't all have the same distributions. And this also shows us that we can't just treat Africa as just one monolith. If you're doing like a clinical trial, it doesn't mean that just because something worked in Ghana, that it's going to work the same way in Kenya or in South Africa. So we need to have all these nuances taken into account when we are working in an African context, especially when we are designing clinical trials. So this is the example I talked about for CYP2D6 and tamoxifen. So I'm not going to repeat this slide, but just to show you that why we need the testing for drugs like tamoxifen. And then this brings me to this slide, which really is a summary of everything I've been talking about. So I know that in this short time, I only had time to tell you about CYP2B6, CYP3F5, CYP2D6 as well. And I also mentioned G6PD before. So this is just the slide that is a summary of many of the genes that currently have clinical guidelines. And a lot of those guidelines are based on what is observed in Europe, but we can apply some of it to Africa, but I think we also need our own guidelines as well. But this is just to show a prediction of how many patients or participants based on the data that we have in Africa. If you were to look at only the genetic data without looking at age, microbiome, and other things, which are also important, but if you were just to look at the genetics, how many people in what would be the proportion of people in Africa that would need dosage adjustment or treatment alterations based on pharmacogenetics. So this is what we show here in the orange. So depending on the drug gene pair, we see that for some of them, very few people need adjustment, whereas for others, we see a lot of people need the adjustment. So this also factors into play when we are trying to implement pharmacogenetics in a clinical setting. So I think based on these predictions, we can see that at the very least for some of the drug gene pairs, we definitely need pharmacogenetic testing, whether it is in a clinical setting or whether it is in a clinical trial investigation. And then I think it would be great if all of the genes here were included in the process. So then, as I conclude, I just wanted to highlight some of the challenges that are impacting implementation of pharmacogenetics. So these are the ones that I thought that stand out. So the cost effectiveness is really key because I know whenever you're working in Africa and you're talking about things like genetic testing, the first question is this feasible? And is this cost effective? I think it could be cost effective in cases like tacrolimus and 3F5, where you're doing a whole organ transplant, because there you can't run the risk of tissue organ rejection, for example. So I think it really depends, or if you're doing something like chemotherapy. So I think it really depends on the context. So for example, if you're treating TB and there's a risk of drug-induced liver injury, you could weigh the benefits against the risks. And I think you'll find that in quite a few cases, the testing will actually be cost effective. But some of the other challenges that exist are that we don't have good electronic health records. So what this means is we need a system to make sure that once someone has received genetic testing in one center, they can show those results to another pharmacist or another clinician if they travel or go somewhere else. The other thing is that we need to bridge some of the knowledge and education gaps regarding pharmacogenetics, because we know that in a lot of education programs, whether it is for pharmacy or medicine, we don't really teach genetics broadly, and certainly not genetics in the context of drug response. So I think we need to do a lot more to try and make this testing a reality in Africa. The other challenge, which is obvious, is the underrepresentation of African populations in, we talked about clinical trials, but also genomics studies in general. So this definitely has to be addressed, and this is part of the work that our group is doing, but we can't do it alone. We need a lot more people joining in on pharmacogenomics in Africa. Then we talked about underrepresentation in clinical trials and the need to consider African ancestry enriched variants and how they might impact the response to both the existing and the new drugs. So I think the question would be, where do we apply this? And I think these preclinical phases, would be a great step to try and first mitigate this risk of adverse drug reactions, especially, and also enhance efficacy before we then go for the clinical trials in this context. So this slide I just got it from this paper because I was just trying to see what are some of the genes that people are including in clinical trials. And as you can see, many of these genes are the ones that I showed in previous slides, which also shows us where we can start from if we are trying to include pharmacogenetics in clinical trials in our context. And I think these also make a lot of sense for our group. And I think I'm looking forward to a lot more efforts in including these genes in clinical trials in our setting. The other challenge I would like to highlight is the challenge of novel or rare variants and their interpretation. So this slide is showing us that in the gray, we are seeing the alleles that we know the function for. So like the star 29 that I talked about earlier and the star ones. But we see that for the novel ones that we found in the current large-scale genomic data, the functional impact is not known. And in many cases, you don't have enough patients that have that variant for you to make a statistically significant association with drug response. So we need novel tools that are able to tell us more about what the functional impact of those variants might be. Otherwise, it will be difficult to interpret them in a clinical setting. And this is work that some of my colleagues are doing. This is a colleague of mine called Husemedin. So he is an expert in using these bioinformatics approaches and these protein models to do molecular dynamic simulations of how different variants will impact how these proteins will interact with their ligands. All different drug molecules. And I think this work can tell us more about the impact of those novel or even the rare variants in our setting. So since we have ISoP online, I think it's also important to talk about pharmacovigilance. Because when you look at VGBase, I think you'll all agree with me that we have very few reports from Uganda that are going into these databases like VGBase. So if you're trying to understand, what is the distribution of, what is the prevalence of ADRs in different countries in Africa, you'll find that there is very little data on that is publicly available. So this brings the question of how can we improve pharmacovigilance, because we need that data to be able to get funding to do more pharmacogenetic studies, but also to understand really what would be the cost, what would be the cost benefit of genetic testing. So without the pharmacovigilance data, it is very difficult for us to show what that cost benefit would be. And so I think it's important to invest as well in pharmacovigilance across different countries and to find more effective ways of recording that data. So here I'm showing a paper where they are doing mobile application to enable patients to also record the effects that they are facing so that these can then be tabulated in more central databases. So I think I'm definitely pleased that APN is partnering with ISoP Africa on this, because then it broadens the efforts to improve it. So yeah, I just wanted to end with this current and future issues. So one of the issues that is there is this concept of a pharmacogenetics passport. Is it possible to do the genetic testing like once and then you have some sort of passport for the patient, so that if they it's like a QR code. So if they go to a pharmacist and they want some medication, they can actually scan the QR code, and then the pharmacist would know that, oh, for this patient, don't give them this medication, or if you're giving them this medication, reduce the dose or increase the dose. So I think that's a clever way, I think, to try and make this accessible, because if you're trying to put this in a central resource, it might be a bit difficult. But if someone has a QR code that people can scan when they go to buy a medication, or if they get a prescription, I think that might be more practical in our setting. So, and then here I just wanted to point out where do we go from here? Like, how do we actually realize the full benefits of pharmacogenomics? So obviously, like for us right now, we are here at the bottom where we are mapping the extent of pharmacogenetic diversity in these drug-related genes. And as I talked about, the next step is assessing functional impact on drug response. But this wouldn't be relevant without pharmacogenomics being part of health policy and health practice. And for that to happen, we need to demonstrate the benefits of pharmacogenetics guided treatment. And this can't be done by just academic researchers or geneticists or computer scientists alone. So we really need partnership with a lot of stakeholders. So that's why I was so excited to present in this meeting, because definitely, working together on this would be vital to then bring everything together and implement pharmacogenetics in health policy and health practice. And then I just wanted to leave you with this slide where they said that do not let the perfect be the enemy of the good. So in other words, there are still some questions about cost effectiveness of pharmacogenomics, but we know that there are some clear examples where, some research, although limited in Africa, has shown us that genetic variation can impact drug response. So I think we need to start somewhere and then take it from there. So thank you so much for listening to me, and happy to take any questions.
Thank you so much, David. This was quite an informative session. Really love how you've walked us through the entire journey from how genetic diversity impacts the clinical decisions, and even given us examples of different drugs and some of the side effects that have been observed. For instance, efavirenz, and also how pharmacogenomics is an important factor when it comes to clinical relevance, other than just age, comorbidities. I think you've done an excellent job, and it's amazing the work that you're doing. And I will just open up the floor to the participants. For those who have questions, please keep them coming. You can use the chat, you can use the Q&A section to be able to do that. I see the participants have also really enjoyed the session. You're getting a lot of claps. So I have a question here from Katu Mutumbi that says, hello, David, could you speak to the role of pharmacogenetics in guiding the use of targeted therapies in oncology? I'm particularly interested in whether your research has explored this space. So if you see it as an area of, or if you see it as an area of future opportunity.
Thank you so much for the question. I think it's interesting because oncology is one of those fields where precision medicine has really advanced. I think about after sequencing the human genome way back in 2001, pharmacogenomics was seen as one of the low-hanging fruits because, it seemed so straightforward that if you know what a variant does to the drug, it's straightforward to test the patient and then adjust the dose or drug based on that. But actually implementing pharmacogenomics hasn't been that straightforward. But when you come to precision oncology, that has actually taken off, and I think that has been way more of it has accelerated a lot more. And us in pharmacogenomics, we have a lot to learn from targeted therapeutics for oncology. But I think to answer your question directly, what might be relevant, I think, is one in the development. So we definitely need So once you've identified these different targets that you can use, we also need to know that the drugs are safe. So because there are lots of side effects to these cancer treatments as well. So this, I think, is where PGX can come into play. So I have not looked at it in depth in my own work, because sometimes also the definition of pharmacogenomics can be very broad. So for us, we are looking at drug disposition a lot more, like absorption, distribution, metabolism, and response. But I think, yeah, we definitely need to venture more into like the oncology thing, because then that makes the definition of pharmacogenomics broader. Yeah, and I think I would love to follow up with the person that asked that question a lot more afterwards. Thank you.
Thank you, David. Katu, I think we will share David's LinkedIn for the follow-up that he has mentioned. And I have another question over here. How are clinical trials currently designed in Africa, and how can we effectively incorporate the nuanced diversity that you talked about in a more sustainable way?
Yes. Thanks for that question. So I don't think I can speak with authority regarding how clinical trials are necessarily designed, but I think we all know the major steps of clinical trials. But what I would love to introduce in this session is GC-ADA. GC-ADA stands for Grand Challenges Africa Drug Discovery Accelerator. So this is an initiative that is being funded by the Bill and Melinda Gates Foundation to try and accelerate drug discovery in Africa. So, basically, people are trying to identify which of the, small molecules could be the next future drugs to use. So I think if we exclude pharmacogenetics in that process, we are likely to repeat the same mistakes that are there currently, whereby drugs have been designed in European settings, or they have only included European participants in trials and have therefore not optimized the dosages or the safety and efficacy profiles to people from other diverse regions of the world. So I think what we are trying to do in our work as well is to partner with GC-ADA, which I've just talked about, to see that we are all moving together in terms of, yes, it's important to do all those processes of lead optimization and preclinical phases of the trials and everything. Now, in that preclinical phase, if you can either do an in vitro assay, for example, you can express some of these variants in different model organisms like yeast, or even some of the human cell lines, and then see how they impact the pharmacokinetic profiles of the different drugs. Of course, you could always do animal studies as well, but I think expressing them first in those model organisms like yeast and HEC293, for example, cells, that would save a lot of money before you then go to the clinical trials.
Thank you, David. Jeremy asks, excellent presentation. Question, if you were to recommend large-scale clinical trial across Africa to understand the genetic diversity, should it be conducted to cover non-communicable diseases treatment or virology treatments?
Ooh, that's interesting. I think both would be the answer, because the non-communicable diseases are also increasing in Africa. So I think both would be the answer to that. And even if you say, let's only do virology treatments. So the example of dolutegravir in HIV treatment has shown us that you might be doing a trial for a virology treatment, but the DTG is being associated with weight gain. So now all of a sudden you now have to worry about the weight gain in the patients, and which will now need a new trial for drugs for obesity. So I think at the end of the day, it has to be both, in my opinion. Yeah.
Thank you, David. Yaz asks, can AI help identify population-specific genetic variants that influence drug response in African populations?
Thanks for that question, Yaz. So yes, definitely. I think this is where we are moving to as well, because we have to, because AI is a strong tool, but the problem with AI is that you also need to feed the AI with data. So if you don't have enough data to begin with, then your AI model may not, you may not learn, it may not learn sufficiently to give you accurate predictions. So there are other approaches like mutational scanning, or what they call deep mutational scanning. So basically they mutate the protein, they create all the possible variants in the protein using saturation mutagenesis. And then they design these assays that can tell you at high throughputs what the functional impact of variants are by expressing them in different models. So that data can then be fed into AI models to help you predict variants in proteins that you did not have in your assay. So AI, I think, as a tool is great, but we need to, first of all, generate enough data that we can feed into those AI models. But definitely a great possibility.
Thank you so much, David. We have a final question from Emily, and she asks, given Africa's vast genetic diversity and its potential to revolutionize personalized medicine, how can the continent position itself to play a more active role in pharmacovigilance and clinical trials for advanced therapy medicinal products, especially considering the current lack of region-specific regulations?
Yes, thanks, Emily. I think this is where I mentioned the GC ADA network, which is the Grand Challenges Africa Drug Discovery Accelerator. So if you just search GC ADA with double D, that's one of the platforms that we can join, I think, to try and enhance our efforts in, especially for the clinical trials. For the pharmacovigilance, I think George will advise better, but like here I showed the other study where they were using the mobile app to try and empower the patients to report their side effects and then record that in a central database. But I think George will have better answers than me regarding the pharmacovigilance. But definitely for the trials and the drug discovery, I would look at what GC ADA is doing. I think they're doing good work, together with H3D, obviously. Yeah.
Thank you so much, David. I don't know if there are any other questions. I don't want to lock anyone out before we come to the end of the session. All right, I can see some participants asking whether the slides will be shared. Yes, the slides will actually be shared after the end of the session, and they will be shared together with the recording. So in case you missed anything, you'll be able to catch on. And David, I think I should just mention that from the participants, they really enjoyed the session. Everyone is commending you for a great job done. I also loved how easy you made the presentation, because I know it was a lot of technical terms, but even for someone who is quite a novice in this space, I was able to get something or two from it. So thank you so much for the amazing job that you've done. And I believe also you've mentioned that with the work that you're doing, you require a lot of hands on deck, and there are so many people, rather so many stakeholders that can be able to support you, even in the role that you are where you're practicing currently, whether you're in the hospital setting, you have a role maybe by reporting the adverse events that you encounter in your practice. So whatever way that you can be able to contribute based on the discussion that we've had today, please do go ahead and do so. And we really hope that in the next few years, we can be able to see improved patient outcomes from the incorporation of pharmacogenomics in our clinical settings. David, I'd like to give you the floor to say something final before we lock it up.
No, just to say thank you again for the invitation, Lucy, and thanks, George, to you too, and great meeting everyone, even though virtually, and keep the webinars going.
Thank you, David. George, do you have anything to add as well?
Thank you so much, Lucy, and thank you, David. This is a great session, and just want to add again that those who are interested in joining us, please follow up on our social media platform so that together we will build a better patient safety culture on the continent. Thank you very much, and nice meeting all of you.
Thank you. Thank you so much, George. Thank you so much, David. We really appreciate taking the time to be here with us today. To the participants, thank you as well for participating. We wouldn't have done this without you. And with that, I come to the end of the session and wish you all a lovely evening.
Thank you. Bye.
Bye-bye.
Thank you. Bye.